You've seen it 100 times on CSI. A detective swabs a crime scene, rushes it to the lab, and within hours, sometimes minutes, they've got a perfect DNA match complete with a photo of the suspect on a giant screen. None of that is real. Welcome to my channel. Today, we're breaking down how DNA analysis actually works in criminal investigations. Not the Hollywood version, the real science. Let's start with what CSI gets wrong. And trust me, it's a lot. First, the timeline. On TV, DNA results come back in hours. In reality, the average turnaround time for DNA analysis is anywhere from 24 to 72 hours, and that's in well-funded labs with minimal backlogs. Some jurisdictions wait weeks or even months for results. Second, the instant match. I'd say TV shows love to display a perfect DNA match with a name and photo appearing on screen within seconds. Real DNA analysis doesn't work like that. The CODIS database, that's the Combined DNA Index system, stores DNA profiles, not names or photos. It's a system of numbers representing specific genetic markers, not a database of faces. Third, the amount of DNA needed. CSI characters can apparently extract perfect DNA from a single skin cell or a decades-old cigarette butt. While modern forensic science has gotten incredibly sensitive, contamination, degradation, and the quality of the sample matter enormously. Not every sample is usable. So if that's what DNA analysis isn't, what is it actually? Let's break down the real science. DNA analysis begins at the crime scene. Forensic technicians look for biological evidence: blood, saliva, hair with roots, skin cells, semen. They use sterile swabs, careful handling procedures, and evidence bags designed to prevent contamination. This is critical because DNA is fragile. Heat, sunlight, moisture, bacteria, all of these can degrade DNA, making it harder or impossible to analyze. Chain of custody begins the moment evidence is collected. Every person who touches that evidence must be documented. Once the sample reaches the lab, forensic scientists extract the DNA from the cells. This involves breaking open the cell walls and separating the DNA from proteins and other cellular material. The extraction process uses chemical solutions and sometimes physical methods like grinding or heating. What you're left with is a solution containing DNA molecules, microscopic strands that hold the genetic code. But here's the challenge. Crime scene samples often contain very small amounts of DNA. A single drop of blood might contain enough DNA for analysis, but a fingerprint left on a surface? That might contain only a few cells, which brings us to the next step. This is where polymerase chain reaction, PCR, comes in. Think of PCR as a molecular photocopier. It takes tiny amounts of DNA and makes millions of copies, giving scientists enough genetic material to analyze. Here's how it works. The DNA sample is heated to separate the double helix into single strands. Then, enzymes called polymerases attach to specific regions of the DNA and build complementary strands. This process is repeated 25 to 35 times, doubling the DNA with each cycle. After 30 cycles, you've got over 1 billion copies of the original DNA. This amplification process typically takes about 2 to 3 hours. PCR was invented in 1983 by biochemist Kary Mullis, and it revolutionized forensic science. Before PCR, you needed bloodstain samples the size of a quarter to get usable DNA. After PCR, a few cells became enough. Now that we've amplified the DNA, forensic scientists analyze specific regions called short tandem repeats, or STRs. Here's what STRs are. Scattered throughout your genome are regions where short sequences of DNA repeat over and over. For example, the sequence GATA might repeat 8 times in one person, but 12 times in another person at the same location on the chromosome. Forensic scientists examined 20 different STR locations, called loci. The FBI's CODIS system uses 20 core STR markers. At each location, you inherit one version from your mother and one from your father, so you have 2 numbers for each marker. Your unique combination of these 20 STR markers creates your DNA profile. The probability that 2 unrelated people have the same 20-marker DNA profile? Roughly 1 in 1 billion or higher. The analysis produces an electropherogram, a graph with colored peaks. Each peak represents a specific STR marker. Forensic scientists read these peaks to determine the numerical values at each location. This process, running the DNA through a genetic analyzer and interpreting the results, takes anywhere from 6 to 12 hours. Once you have a DNA profile from crime scene evidence, forensic scientists compare it to option 1, a known suspect's DNA. If police have a suspect, they'll collect a DNA sample, usually a cheek swab, for comparison. If the profiles match at all 20 markers, that's considered a positive identification. Option 2, the CODIS database. CODIS is the FBI's national DNA database system, which contains over 14 million offender profiles and over 4 million arrestee profiles as of 2024. When a crime scene DNA profile is entered into CODIS, the system searches for matches. If there's a hit, investigators receive a notification that a profile in the database matches the crime scene evidence. Then they use traditional investigative methods to confirm the match and build a case. Important note, CODIS only stores the 20 STR numbers, not names, not photos, not genetic information about health or physical traits. It's purely an identification system. Here's where it gets complicated. Not every sample is clean and clear. Nice mixed samples. Crime scenes often yield DNA from multiple people. If a blood sample contains DNA from both the victim and the attacker, forensic scientists must use statistical methods to separate the profiles. This is mathematically complex and can lead to interpretation challenges. Degraded samples, old evidence, evidence exposed to environmental factors, or evidence from challenging sources like bone or teeth might yield partial DNA profiles. Scientists can still get information from these samples, but the statistics become less certain. Contamination. This is the nightmare scenario. If crime scene investigators don't follow proper protocols, DNA from police officers, lab technicians, or even forensic scientists can contaminate the evidence. This is why proper collection and handling procedures are absolutely critical. When forensic scientists say this DNA profile matches the suspect, what they really mean is the probability that this DNA came from someone other than the suspect is approximately 1 in 1 billion, or whatever the calculated statistic is. These statistics are based on population genetics, how common or rare specific STR combinations are in different ethnic populations. The rarer the combination, the stronger the match. This is why you'll hear terms like random match probability in court testimony. DNA evidence is presented as statistical probability, not absolute certainty. Let's look at how DNA analysis has actually solved cases. The Golden State Killer, 2018 Joseph DeAngelo committed at least 13 murders and 50 rapes in California between 1974 and 1986, then disappeared. For decades, investigators had DNA from multiple crime scenes but no match in CODIS. In 2018, investigators used a new technique, forensic genealogy. They uploaded the crime scene DNA profile to a public genealogy database called GEDmatch, found distant relatives, and used traditional genealogy research to build a family tree. That led them to DeAngelo. His arrest demonstrated how DNA technology continues to evolve. The Innocence Project DNA analysis has also exonerated over 375 wrongfully convicted people in the United States as of 2024. In many of these cases, DNA testing on old evidence proved that the person convicted couldn't have committed the crime. In some cases, DNA analysis identified the actual perpetrator who had gone unpunished for decades. This demonstrates both the power of DNA evidence and the limitations of other forensic techniques used before DNA analysis became available. Modern cold cases. Forensic genealogy has now solved dozens of cold cases from the 1970s and 1980s. Cases that seemed unsolvable because the perpetrator wasn't in CODIS are being cracked by tracing DNA through distant relatives. This technique raises privacy questions we're still grappling with as a society, but it's undeniably effective. DNA analysis is incredibly powerful, but it's not perfect. Here are the limitations you need to understand. Transfer DNA. Your DNA can end up places you've never been. If you shake someone's hand, you transfer skin cells. If that person later touches a crime scene, your DNA could be there. This is called secondary transfer, and it's a real problem in forensic science. Just because DNA is present doesn't automatically mean that person committed the crime. Lab errors, while rare, mistakes happen. The Houston crime lab scandal in the early 2000s revealed widespread problems including contamination, mislabeling, and poor protocols. Hundreds of cases had to be reviewed. Quality control in forensic labs is critical, and not all labs meet the same standards. Partial profiles. When DNA is degraded or limited, scientists might only get results from 10 or 12 markers instead of 20. This reduces the statistical power. A partial profile match is much weaker evidence than a full 20-marker match. Database limitations. CODIS only works if the perpetrator is in the system. As of 2024, the database contains about 14 million offender profiles, but there are over 330 million people in the United States. Most people are not in CODIS. Let's recap what you've learned today. 1. Real DNA analysis takes 24 to 72 hours minimum, not minutes. The process involves collection, extraction, amplification through PCR, STR analysis, comparison, and statistical interpretation. 2. CODIS stores numerical DNA profiles, 20 genetic markers, not photographs or personal information. It's a matching system, not a criminal database with photos. 3. DNA evidence is presented as statistical probability. When scientists say there's a match, they mean the probability of it being someone else is astronomically low, but not zero. 4. DNA analysis has revolutionized criminal justice, solving cold cases and exonerating the innocent. But it's not infallible. Transferred DNA, lab errors, and partial profiles are real limitations. 5. New techniques like forensic genealogy are expanding what's possible, but they also raise important privacy questions our society is still working through. The next time you watch CSI, you'll know what's real and what's Hollywood. Real forensic science is slower, more complicated, and way more interesting than TV makes it look. If you learned something today, hit that subscribe button. We're breaking down forensic science every week, from fingerprints to digital forensics to wrongful convictions. Want to learn more? Check out the sources in the description. We've linked to the FBI's CODIS information page, the Innocence Project's DNA exoneration database, and scientific papers on forensic DNA analysis. Drop a comment. What forensic science topic should we cover next? Fingerprints, blood spatter, digital forensics? Thanks for watching. Remember, not CSI, real science.